We all know that we're facing a climate emergency. It's on the news every day, it's in all kinds of scientific reports all the time. You can look out the window in the middle of January and see how sunny and warm it is. At least I can here. But we don't usually talk about this too much in neuroscience. Neuroscience isn't really concerned with climate change. That's not to say we don't care about it, but it's just not part of what you think about when you think about cognition and moment-to-moment decisions. Climate change takes place over a huge amount of time. But there is this question that climate change is always wondering about, at least from a biological perspective, of how can animals deal with climate change? How can we adapt? How can we change? Where are the breaking points? What's the limit of that resilience? We don't really know. We have no good answers to these questions. But again, it's not something neuroscience really studies, or do we? So in the paper I'm going to tell you about today, recently published in the Journal of Neuroscience by Eve Marder and one of her graduate students, Mara Rue, they report on some of the kind of serendipitous events that led Marder to being an accidental climate ecologist. Over the course of her several decades studying the somatogastric ganglion found in crustaceans, she's traced out in exquisite detail how particular neurons in this structure produce a beautiful triphasic rhythm of 3 different types of neurons that fire 1, 2, 3, 1, 2, 3, 1, 2, 3, and so on, at a period of about 1 to 2 cycles per second. She never expected that in doing this she was going to end up studying climate change, but that's exactly where she ended up. And in this really phenomenal synthesis review article, her and her graduate student traced through all of the various trials and tribulations that kind of led them along this path to eventually realize that actually they were studying climate change. So the quick punchline is that crabs can actually adapt phenomenally quickly to changes in ocean temperature. But come along and let's get into a little bit more detail. Hi there. My name is Adam Dede and welcome to a Neuroscience Journal Club, where I try and bring you a weekly update on the world of neuroscience by talking about a recently published peer-reviewed article. So, today we're going to talk about the somatogastric ganglion of crabs and lobsters. Now, if you're like me, when I first heard this as a topic for a talk, I really thought this was going to be a pretty boring one. But it turns out that Eve Marder's work is really phenomenal. It's elegant, it's amazing, it gives you insights into things far beyond crabs and lobsters. So hopefully you'll stick with me. Now first, got to introduce you to what it is that we're studying. So the somatogastric ganglion is a set of neurons in the crab and the lobster that controls the motion of muscles in their digestive system. And it's not really particularly important to understand what those muscles are trying to do, but what is important to understand is that there's this really amazing set of 3 different neurons that fire in sequence, PY, PD, LP. So that's what these 3 types of neurons have been named, PY, LP, PD. And you can see in the figure here, they always fire in that order. PY fires a burst, then PD fires a burst, LP fires a burst, then PY fires a burst, then PD fires a burst, and so on. LP, PY, PD, LP, PY, PD, LP, PY, PD. Now, how it is that the timing of this really cool oscillator comes about has been the subject of Marder's research for decades. And in the early days of trying to study this oscillation pattern, it was thought that if you could just get a real handle on exactly what the wiring diagram looked like, on how these different types of neurons were wired together, you'd get all the way there. You'd be able to understand, oh, wow, amazing. We now understand the way in which these oscillations are produced. And hopefully, in understanding how these oscillations are produced in this relatively simple circuit, we can gain an insight into how oscillations are produced in more complicated circuits. So here's the wiring diagram. So cool, this is going to be a really short video. You can obviously see exactly how the oscillation is produced, right? No, of course not. And neither could any of the researchers who were working on it. Everyone was still stumped. Getting the wiring diagram does not tell you how the oscillation is produced. So the next thing that Marder and her team did was they went to computational work, and they tried to think, okay, well, each of these types of neurons, they all have many different pores or ion channels in the membrane of the neuron, and each of these ion channels has a different electrical property in what types of ions it allows to pass in and out of the neuron. So if we can model a neuron as a collection of these ion channels and then get some kind of a mathematical estimate for the net conductivity of the neuron, how electrically conductive it actually is, maybe we can model a set of neurons that's capable of producing this triphasic rhythm of these three neurons firing. And then when we figure out what is the model that can produce that rhythm, we'll know something about what exactly is going on that allows for this exquisite timing. So Astrid Prinz, working in Marder's lab in the early 2000s, came up with 20 million different biologically possible or plausible models of these three neuron types, and wired together as they are known to be in nature, and checked in the computer to see whether or not any of these 20 million would be capable of producing the triphasic rhythm. Now, it turned out that 400,000 of them were able to do it. So, yeah, 400,000 out of 20 million, it's a little less than 5%. That's not that many, but at the same time, 400,000 is actually a huge number. And what's more, it turned out that it wasn't as if these 400,000 existed in a very similar space to each other. In fact, the models were very different. So in this figure that I'm putting up now, what you can see in the top half is two different sets of simulated data from the models showing the triphasic rhythm, that typical activity pattern of three different types of neurons firing in sequence. And then in the bottom half, what you're looking at down there are the different conductances that went into the model. Now, it's not important exactly what those conductances are. What's important to realize is that they're very different. So we can see that in some instances, certain patterns of conductance can produce this one set, this rhythm. And then over here, we see totally different sets of conductances that produce, oh yeah, exactly the same rhythm. So, okay, there's massive variability. It's a degenerate case. There are multiple valid solutions to the one problem of how do we get this triphasic rhythm. Okay, now you might be wondering, that's all well and good to do in a model, but I want to see real data. I want to see actual data from the animals. So, it took them like 15 years from the publication of Astrid Prinz's work before they actually managed to do this. But they did single cell level PCR to count the number of RNA copies of different ion channels. And in this way, they can actually count on a single cell level how many ion channels of different species there are in that single cell. And what they found in looking at samples taken from many different crabs was that from one crab to another crab, there was a 2 to 6-fold difference in the concentration of different ion channels across the individual crabs. And this was true across a wide range of different ion channel types. So this is pretty crazy. This is the same type of variability that we saw in the model now in real data. There is actually massive variability in the ion channel concentration from one animal to another. And so that tells us then that, yeah, in nature we actually do see this degenerate case. We see these multiple solutions, and yet all of these animals are producing the same triphasic rhythm, even though they have vastly different ion channel conductances in their cells. So what's going on? How can we have so much individual variability in terms of ion channels, but we have no individual variability in terms of the outward activity of the circuit? So this was what led Marder to start looking at perturbations. What happens when you challenge the system with changes in its environment? And one of the easiest to do and most most logically immediate is to just change the temperature. So she started looking at what happens when you change the temperature in which the recordings are collected. And it turned out, unsurprisingly, that if you crank the temperature up high enough, usually these recordings are done in about 11 degrees Celsius. And when you crank them all the way up to about 28 degrees Celsius, you see a crash. The triphasic rhythm ceases to be produced properly. And two interesting things can be drawn from this. One, all the way from 11 degrees up to 28 degrees, different individuals with vastly different ion channel distributions are all managing to handle the perturbation of temperature. But more interestingly, at the high temperature, what they found was that each individual crashed in a unique way. So here we're taking a look at the activity of these three neuron types at a very high temperature. And what you can see is that although at lower temperatures they all exhibited the classic triphasic rhythm, now we can see at high temperature that they're crashing in unique ways. Some of them we see massive increases in firing, sometimes we see them go completely silent, but different cells or different individual crabs are producing different changes when they hit this crash point. So there's a revealing of the individual differences that we knew were going to be there from the modeling work and from the single cell RNA counting. Okay, so that's cool, and that's something that Marder's lab has continued to pursue ever since they started seeing this phenomenon. But what they found just by accident as they were doing this was, so back in 2006, the first sample of crabs that they looked at with these temperature perturbations, they found, as I said, about 28 degrees Celsius was the crash point. But then in 2012, they happened to find that they had crabs that exhibited a crash point up in the mid-30s, so a big difference. And then back in 2016, they were back down, showing crash points in the upper 20s. And finally, in 2021, they had crash points up in the mid or even the upper 30s. They had one animal whose crash point wasn't found until 39 degrees Celsius, which is really pretty hot water. So how is this the case? How are they seeing such variability one year to another? And you can see here, this wasn't just individual cases. You can see the distribution of crash points for animals that were sampled in 2016 versus animals sampled in 2021 is really vastly different. And it's important to note, they're getting wild-caught crabs. These crabs are generally 5 to 7 years old. They're robust, they're adults, they've survived. They've made it through many seasons, they've weathered the tide. They're out there in the elements. These are strong individuals. So why is it that we're seeing such a massive difference in the crash point temperature? So Marder and her colleagues went at this point to get publicly available ocean temperature data for the area just off the coast of Massachusetts where their crabs were collected. And what they found was that right in sync with the changes they were seeing in the crabs' crash points from 2006 to 2012, back down at 2016, and then up in 2021, these correlated really well with changes in ocean temperature. Basically, if ocean temperature was hot, then they ended up with crabs that could handle heat. If ocean temperature was cold, they ended up with crabs that couldn't handle the heat so well. Effectively, they were looking at a fast reaction on a population level to climate change. So this is really cool, and it gives us a few super interesting conclusions. So one, we don't see individual differences until we expose the animals to stress. Keep in mind, all the way up until the crash point, we don't see any difference. We know that there's a difference there from the modeling work and the mRNA counting, but the difference doesn't show up in the circuit activity until we hit that crash point, and that's where we see unique types of crash. A third point that's really interesting to think about is that evolution has to be selecting for this kind of resilience. Now, the reason for this goes back to that modeling work that we talked about earlier. In that modeling work, there's 400,000 different model simulations that are capable of producing the triphasic rhythm. But importantly, there's almost none, very few, and it's very hard to find them, that are capable of handling any kind of perturbation in temperature at all. What this tells us is that evolution is selecting for resilience to temperature perturbation. If it weren't, then we wouldn't see many animals that would be able to handle it, but we see all of them are able to handle it to at least some degree. Keep in mind, a 28-degree ocean is not something that happened off the coast of Massachusetts. And then the last interesting conclusion here is that the adaptation is fast. And this is something that I don't think many people would have expected. I certainly wouldn't have expected it. The fact that we see from year to year changes in ocean temperature result in changes in the animal's ability to handle temperature. Now, what this means is really hard to interpret. It raises a lot of questions. Like, for example, does this kind of resilience come at a price? Is it the case that animals are becoming resilient to temperature at the cost of resilience to pH or potassium concentration? And those are actually studies that Marder's lab is in the process of trying to look at. How does interaction in environmental changes produce different types of resilience? Another question that's important to ask, does the overall gene pool of the crabs end up changing as a result of being exposed to these kind of temperature perturbations? Is it the case that we're seeing an enrichment of temperature resilience, but maybe there's a loss of genetic diversity that might have existed at the lower temperatures but is going to be gone at the higher temperatures? What does that mean for the future integrity of the overall population? It's unclear. Third, where's the limit of this resilience? Obviously there is one. If you put a crab in boiling water, it's not going to survive for very long. So where is that limit? We know that the crabs seem to be able to function just fine in Marder's experiments up to, maybe somewhere in the low 20s. But where's the line? How much resilience is there in the system? And then last, bringing it back to us, as we like to do as humans, what does this mean for humans? Human environments are changing rapidly, and as we talked about in the video last week, changes in human environments can radically change brain activity. So is it the case now, thinking about this in the context of this study, that human brain activity actually changes not over the course of a lifetime, but on a much smaller time scale from year to year, or even maybe it's month to month, who knows? What does this mean in terms of kind of what you might think about as long-term brain states, long-term nervous system states? Clearly the crabs are showing us that there is such a thing as a long-term nervous system state, a state that can be adopted and held for months at a time in reaction to an environmental change. Is that something that humans exhibit as well? Obviously, more questions than answers almost on this one, as is true of much interesting science, but hopefully you enjoyed. I'd love to hear your comments or interpretations down in the comments section below. If you enjoyed the content, please consider supporting me on Patreon, where you can have input into what videos or what articles I review in future videos, and also get access to videos before they're posted on YouTube. In the meantime, please like, subscribe, and I'll catch you next time.